So I will talk about a clinical phenomenon which is known as sleep benefit. So that I will introduce with the words of a patient of mine who I met when I was working in Switzerland, and she said that there were some days in which she woke up in the morning and it was like if Parkinson's disease has disappeared. So she said, I experienced as having been cured from Parkinson's disease at the point that I forget sometimes taking my medication, but then the miracle dissolves after 1 hour or 2 hours, and so I feel off. But this phenomenon was really interesting for me because it's something that is not very well described and well explored in the scientific literature, but that the patient described very well, some of them. May I do a quick survey? How many people with Parkinson's disease are in this room right now? Okay. And how many people have experienced a sort of dramatic improvement in their movement upon waking, either in the morning or in the afternoon after a nap? Okay, great. A bunch of people, I'd say maybe 30% in this room, roughly speaking. It's something that is also found in the literature. According to some authors, it is a phenomenon touching 30% of the people, according to others, up to 72%. It's typically variable, so it can change a lot from a day to another, and is transitory. So this is why also it's quite difficult to explore this phenomenon. In the literature, there are a few open questions, because there are some researchers who even question if it is a real objective clinical phenomenon or if it's just a patient experience. And they argue that it's not measurable because there have been many attempts from different groups worldwide to measure it. And according to someone, it's not possible to measure this phenomenon. And other also question that it's really sleep dependent or if it's something that it's just due to rest. So to try to answer these simple questions, when I used to work in Switzerland, in Lugano, I set up with my collaborator a study in which we invited many patients with Parkinson's disease at the hospital to sleep overnight with doing the sleep study, the one that people argue they cannot sleep when doing. But our patient sort of slept during this time, and we made a clinical assessment of their motor performance before they went to sleep, then on morning waking, and then we repeated an assessment after 1 hour after they took their medication from Parkinson's disease. And during this assessment, we made some videos in order to be able to measure something else from the video and not just with our eyes. So this was the setting of the polysomnography. It's the way we study sleep. We have some electrodes on the head, some electrodes near the eyes, some electrodes on the muscles. These are the mandatory channels that we are studying to characterize sleep and wakefulness, and I will give you now an example of a page of wakefulness when we start recording. In the first two lines up there are the eye movements, so they are pretty flat here. Then we have the electroencephalogram, so the activity of the brain, and here we have the muscles. And this is we see at the EEG, the electroencephalography, that the activity is hustle and bustle. It's like being at a market when everybody shouts, talks, etc., but we cannot recognize a specific activity. This is what we call, in EEG language, desynchronized activity. And it's typical of wakefulness, but not only of wakefulness, because when we then dig into sleep, we get to a phase, which is the REM sleep phase, that is represented here in red, in which the activity is still desynchronized. But there is another stage of sleep which comes before the REM, rapid eye movement sleep, which is called the slow wave sleep, which is represented here in blue. And this part of sleep is where we sleep more deeply, and when our brain and our body are recovering from the activity of the day, from their energy expenditure. This is an example of a page of REM sleep, rapid eye movement. It seems that when we are in REM sleep, we are chasing our dreams, so we are following our dreams with our eyes, and we see here in blue that the eyes are moving, basically. The EEG is still desynchronized, so hustle and bustle activity, and the muscles are paralyzed. This is what usually happens in REM sleep. Then, when we are in the slow wave sleep, we have a synchronized activity. All the neurons of our brain are singing together like at a concert, like a choir. And in this case, we can see a pattern of activity at the EEG. This is the example. And provided that when we sleep, we have different cycles, so we repeat going to deep sleep, then REM sleep, then deep sleep, then REM sleep, at least 4 or 5 times every night. We can then trace, in measuring with mathematical methods, we can then see that our brain is producing slow wave activity 4 times at least during the night, or 5 or 6, depending on how long we sleep and how many cycles we do. But basically, this is the power of the slow wave activity that we can extract from the sleeping electroencephalogram. And we can then derive a slope, so a decay rate of this slow wave activity, which is a good correlate of our energy recovery process that's happening during sleep over the night. So coming back to our experiment, so we measured the activity, the motor performance at bedtime, on waking, and also we measured what was happening during sleep. So we measured this slow wave activity decay during sleep, and we tried to see if there was a connection between how the patient improved in their movement from bedtime to morning and this slow wave activity decay of non-REM sleep. This was how we looked at this patient. I was much younger, so it took some time in analyzing this data and getting them. And basically, we had some video recordings from our patient while we were doing the MDS UPDRI scale, so the task that you always repeat every visit, so finger tapping, hand gripping, and pronosupination of your hands. So we took these 3 movements of the UPDRS, and then we employed a machine learning approach with artificial intelligence to extract what we call some features. So it's exactly the same approach that Timothy explained this morning. And basically, instead of taking a score from single movements that we were observing, we took some features. So we extracted some parameters that help us identify the movement characteristics. These are frequency, amplitude, speed, and periodicity. It's not a comprehensive assessment of movement, it's just an angle from which we were looking at the problem. But this is a way to decompose a complex reality into more simple questions. For instance, frequency is a way, I can show you what different frequency of a movement like a clap can be. This is one example of a movement which has a certain frequency, and this is another one. Okay, so two different frequencies. In terms of amplitude, this simple movement can be like this or like this. Okay? In terms of speed, we can have this or this. And in terms of periodicity, we can have a periodic movement or a non-periodic movement. For instance, we can have this, or this. Okay, so we try to apply this approach, so extracting some features on the three tasks of the UPDRS scale. And now I show you some examples. This, for instance, is an example in one patient, so how the frequency of the movement that was captured by the artificial intelligence based on the video. So on the hand gripping task, so this open and closing the hand, and we see in red we have the bedtime spectral frequency from the movement, then in green what's happening on waking after sleep, and in blue it's after the medications. So we see that the pattern is changing basically already spontaneously before taking the medications. And then if we consider the amplitude, this as a group level in 28 patients, so we see that at bedtime, so from bedtime to wake time, there was a dramatic improvement in amplitude, the amplitude of these movements, and then there was an extra increasing amplitude after the medications. And so was in speed, and here the different boxes indicate different fingers of the open and closing the hands task. We see the same pattern, an increase in speed from bedtime to waking, and then an extra increase after the medications. And lastly, for the periodicity, we see an example here in one patient where we see the periodicity spontaneously increased from bedtime to waking, and then it was smoother after medications again later. So then we came back to the EEG and derived from the real data this slow activity decay, and it was a lot of pre-processing time, as Tiago knows, so a lot of time to do to prepare the data, and then we could correlate this data, so the energy recovery taking place during the night with these four motor parameters. So this is to understand these figures, we see that when there is a line that points towards up, it means that there is a connection between the two variables. So this is a correlation. There is a good correlation. There was a good correlation between the slow activity decay and all the four parameters at the group level in 28 patients. So this is just a preliminary study, but it's helping shedding light on a complex problem, which is the one of sleep benefit. So it's an angle from which we can look at this phenomenon, and it's very encouraging because we could objectively measure this improvement that occurs spontaneously after sleep, and we could measure it, and we see also that there is a good correlation with the very important sleep process that occurs during sleep, which is the energy recovery. So the next step is that I'd like to move forward and set up this line of research in Oxford as well. So I need your help. So I need the help of patients or relatives that are interested in working with us as advisors. So if there is anybody interested, they can drop me an email, or I can wait outside at the end of the day to get your names if you would like to help us as advisors, and for sure also as patients if we eventually get the funding for this project. Thank you so much. You're so listening.